We are going to have our third presentation of the day. It is my honor to introduce our next speaker, Meeya Odell. She is also a fellow bird nerd, and she comes to us from many different places. Most recently, she's established in Volcanoes with her family. And she also brings a unique perspective of her Japanese heritage to the work that she does and the work that she's interested in when it comes to avian fauna. So I think that this next talk, you'll find it's a very interesting survey of different things that you can do to assist with avian, with avifauna and the really important work that comes with the conservation of both our forest birds and our waterfowl. So with that, I'd like to invite Meeya up to the stage.
Hey everyone. Is that better? Can you hear me? Okay. Hi everyone. So I'll be talking about my internship experience at Hawaiʻi Volcanoes National Park through my involvement looking at avian malaria transmission along Hawaiʻi ʻAmakihi and Nēnē surveying. And when I first started this master's program, my initial objectives were to improve my writing and data analysis skills. Going into this program, I had experience with both, but I just wanted to continue improving on it. I have experience. I had a good amount coming into it, but I just wanted to expand on it further, but most importantly, connect to study birds. Was Hawaiʻi Volcanoes National Park, but I worked at 2 agencies during this time period. And the park is 2 of the 5 volcanoes on Hawaii Island, and both are located within this park. Mauna Loa being 3,500 meters high in elevation, making up about half of the area. And Kilauea, on the other hand, is the youngest among them, and the summit is about 1,240 meters in elevation. And within the park, I first worked with the USGS Pacific Island Ecosystems Research Center, or PIERS, and the National Park Service's wildlife team. And within these 2 organizations, it was mostly in the main area of the park, looking at Hawaiʻi ʻAmakihi and forest birds and their disease dynamic and Nēnē survey. This work was at Ainohu Ranch, and either through bird banding or the surrounding area around the ranch house for Nēnē surveys. But for Nēnē surveys, it also involved going to Volcano Golf Course and Hilo Military Camp and other parking lots along devastation and chain of craters. Introduce birds a little bit. Hawaii's island chains, geographic distance from land masses, and topographic variation leads to due to its volcanic origin, leads to different climate zones and biodiversity, leading to the adaptive radiation of a lot of organisms, including our forest birds. These birds mostly comprise of honey creepers, but there are a few species of thrush and 2 raptor species. And these honey creepers are a result of the arrival of Eurasian rose finches of about 5.7 million years ago. Forest birds are important for our island ecosystems, but as early as 1890s, their populations are experiencing great decline due to both naturally occurring and human-caused events, and introduction of invasive species and pathogens contribute to this decline further and threaten their. Are the birds found within the park? The first focal species of my internship, being the Hawaiʻi ʻAmakihi on the left here, are listed as least concerned because they have some resistance to one of the pathogens introduced, which is the Avian malaria. And they have a stable population within these lower elevation areas because of their resistance to the disease. But a lot of these other forest birds are not able to have this resistance. So they're more threatened or facing extinction as a result. And Hilo and on the bottom row is ʻAmakihi, Akepa, and Hilo. Forest bird habitats once range from sea level at 0 meters to high elevation areas, so everything past 1,800 meters. But these mosquitoes are vectors for avian malaria, the bloodborne disease spread by specifically the southern house mosquitoes through their sour ducts transmitted when they're blood feeding. But it's only the female mosquitoes that blood feed prior to laying eggs. And they typically bite birds while they're sleeping on exposed skin, for example, their legs or around their eyes and around their beaks. So photographed here explains how an infected mosquito, when they're blood feeding, could transmit the disease to an uninfected bird, but it could also be the other way around, where an infected bird passes it to a uninfected mosquito, who will then transmit it to another uninfected bird, infecting them in the process. Are transmitting the disease is often associated with elevation in temperature, and as climate change continues and alters disease dynamics and threatens their populations, and this earliest trend of mosquitoes moving up in elevation and further reducing bird habitat range and limiting their space is a trend that's been recognized since 2013. But at the moment, there's no vaccine or cure for avian malaria, and that doesn't monitoring and studying how it is spread and understanding mosquito prevalence and how it is affecting birds. With all this in mind, the National Park Service and USGS created the Avian Disease Dynamic Project. So it's a partnership project between them studying avian malaria spread and how it affects lower elevation areas like Hawaiʻi ʻAmakihi. And my involvement ranged from fieldwork, laboratory work, and data analysis. Participating in bird banding and mosquito trapping, and collecting samples during both these events and taking them back to the lab for DNA extraction and blood smear analysis. But my main deliverables throughout this time was data analysis, looking at both the blood smear data set with the Hawaiʻi ʻAmakihi of whether they carry diseases or not, along with information about the individual who the sample was collected from, and phenology for Nēnē and Meeya, which are two of the several sources of food for Hawaiʻi. As for the field work aspect of things, for my involvement in bird banding, this involved placing mist nets up in the morning after arriving at the bird banding station and checking approximately every 20 to 30 minutes to determine whether a bird is tangled in the net. And if they are, we would gently detangle them, place them into one of these cotton bags, and take them back to the bird banding station. And in the right middle photo, you see these bags hanging up on these clotheslines, and each of these bags has a bird, and that it just shows that if there are more birds and people were able to process it, they kind of just stay on these lines until somebody's ready. And after taking them out of these bags, you would take various measurements for their wing length, their tarsus, and beak length, write down on the data sheet along with their band number. And if they don't have a band, you would attach one. And after that, you would collect blood, colloidal, and other samples. As for the blood samples, this requires folding their wing out to locate a vein. So you very gently spread their wings a little bit underneath their human bird equivalent of an armpit, basically, and allow the blood to drip into a capillary tube. And you tap this capillary tube on a microscope slide and create a blood smear. And both these two forms of for further analysis and part of the blood smear data set that I was looking into there, which I was speaking about. Although this process is mainly for Hawaiʻi, it's also white-eyed warblers. They're an introduced species, but they're considered reservoirs for avian malaria. And because they are also susceptible to avian malaria, but they're not dealing with population decline, and because it's not fatal to them. But because they're reservoirs, it is getting passed on to Hawaiʻi indirectly and contributing to their decline. So other birds, during these bird banding days that you meet include the the last days that we went, but occasional finches, and I've also heard of somebody catching a cardinal at one point. But I never got to see cardinals, and that was unfortunate for me, but I was glad that I got to see them and meet white-eyed warblers and ʻAmakihi and Avian in person and up close. And when this disease dynamic project ended, I was able to help take down these misnet posts and take them back to the USGS office. And these blood smears that I was talking about collected at bird banding sites, they're really important for understanding Hawaiʻi resistance or immunity to avian malaria. So by looking at these blood smears under the microscope, you're able to count and identify the different white blood cells and red blood cells, and even see whether that individual had and the machine will tell you whether that individual had malaria or not. Here's an example of what a blood smear will look like under a microscope. And to kind of explain this a little further, there's 5 types of white blood cells. There's the mononuclear cells, including the lymphocytes and the monocytes, and the granulocytes, including the heterophils and eosinophils. And then the 5th one, the thrombocytes, are not counted in this data set, but there is noted and recognized. But if they're surrounding each of these white blood cells in the photographs. And I received the blood smear data set basically completed after participating in different aspects of data collection. I looked at the blood smear data set, just looking at questions that you could answer through this data set, including this figure that first ones that I created. And this one shows how infection status among different age groups. And age groups you can determine by the development of the plumage of the birds. So you can see in these 2 examples, one's older and younger, and you can tell based on the brightness of the feathers and appearance, but you can also see up close how worn they are and whether they have fall marks spreading out or not. And by looking at this figure, you can kind of see that older birds, the ones in this represented in this bright green or developed plumage, they have higher prevalence of malaria. So it kind of indicates that the cases of avian malaria increases among the. So my data analysis for the blood smear data set is still ongoing, but this is just one of the things that I've looked at so far. My next steps is to look at the rates among the birds, depending on the number of red blood cells and white blood cells that they have. And for my participation, I got to experience mosquito trapping and to understand mosquito population and presence among this lower elevation area like Idaho Ranch. And this photograph is an example of a Culex mosquito, which are the vectors for avian malaria. Traps and transporting these little containers back to the lab, we will place them in freezers to make sure they're essentially dead. So a lot of them were Culex, but also the others were Aedes mosquitoes. So you separate them based on the species, and then you take the Culex mosquitoes because they're the carriers of avian malaria and dissect them under the microscope, and then they undergo tests later to determine the area. The that I got to analyze and contribute to was the phenology one for ʻavian. And Hawaiʻi ʻAmakihi are, even though they're generalist species, nectar from these two flowering plants are part of their diet. So looking at the availability and the bloom trends of these two plants and flowers are really important to maybe potentially get an idea of food sources and how that helps fight malaria. I created for Mamane, you can kind of see that there's two bloom periods within two peaks. Let's show that there's 2 months with no bloom between the 2023 and 2024 periods of the data collected, but within 2023, there's 6 months of bloom and 9 months of bloom for 2024. For here, there is only one of the data set I analyzed, but their bloom period is much shorter than Mamane. They only have 4 months for both of the years, 4 months each, but there's 8 months with no blooms. So after my participation with the Avian, the disease dynamic project, work aspect of things kind of slowed down. Even though I am contributing and still analyzing the data set, I need more field work hours, and participating in Nēnē surveys was something I was interested in doing still, so I was able to continue with that. And Nēnē is technically a water bird, but if you're being more specific, they are considered a waterfowl. And water birds, including Nēnē, are culturally and ecologically significant for their habitats, but their habitats vary more than these forest birds because they can live both on marine and terrestrial habitats, but they also include migratory birds and shorebirds and seabirds. But there's only 6 endemic water bird species in Hawaii compared to the 84 endemic forest birds. And within the park, there's been Indian populations, but other water birds include the Hawaiʻi and ʻuaʻu. But Nēnē is still an exception among these water birds because their habitat preferences vary even within this group of birds. And it includes wetland habitats, lava flows, and grasslands, and they can live in high elevation, and sometimes they've even been noted to travel between islands. The Nēnē is one of 6 endemic geese, but the other 5 are now extinct, and they can only be described from fossil records. But within Hawaii Island and Big Island, only 25% of the Nēnē population resides here, compared to the 61% on Hawaii and 13% on Maui. So it is very important to continue to monitor these threatened species, especially as an island with a smaller population size, but in total, the estimated population is about 4,300. Next slide, I'm talking about threats to the Nēnē population. So there is a dead bird, just a heads up. So to talk a little bit more about the threats, Nēnē aren't anywhere, but disease like toxoplasmosis and avian botulism threaten and often lead to fatalities for goslings. But regardless of development age, their biggest threats are predation from mongoose, pigs, and cats. Also very prone to road collisions, unfortunately, because their diet consists of grass and berries, and they like to forage on sides of roads or there's been 118 Nēnē fatalities from 1918 to 2023, and mortalities peaking during gosling season and when they're molting. It's really important to continue monitoring these threatened species, like I said earlier, and surveying during breeding season includes Nēnē have a 9-digit fish and wildlife code band on their one leg, but on the other leg they have a color band, and then figure out who they're spending most time with. After that, you have to determine whether they're nesting or not, and if they're nesting, you have to pinpoint the location on the GPS and place a orange tape around it, and within this circle, this little green circle, you probably can't see it, but there is a nesting female Nēnē sitting on a nest. And then after that, you have to mark down what stage of development they're at, whether they're nesting, whether they hatch, the number of goslings. And another aspect of the survey is determining whether they're male or female based on characteristics of each. So for females, you can kind of see in this photo, but you can look at their FAT score, which is when their underside protrudes, and this is a 3.5 out of 4, so they're pretty close to laying eggs. You can also look at root patch, which is where on their chest they lose their feathers to allow them to make contact with the egg directly with their chest. But if parents are doing their jobs adequately, the female would typically sit on nest for several hours at a time, except the nest breaks. And during this time, the male would keep watch, make sure there's no intruders and predators. But if there are, they will come at you and hiss and chase you. Like this male, he came towards me, even though I was very, very far away, and he came chasing after me and screaming and hissing at me with his mouth open. You have to mark down what stage of development they're at using this reference sheet. But at the moment, this year's Nēnē season within the park ended a little bit earlier than anticipated. So my participation is shifting towards Nēnē monitor and placing and helping out to continue protecting Nēnē populations, especially during breeding season, but also outside of this time frame. One way is with this New Zealand activated trap, which is a kill trap. So if a smaller predator tries to eat the bait, it will kill them instantly. But we also use live traps, but I did not include a picture of that. Another way to make sure we continue protecting Nēnē populations is through making sure people are not feeding the birds. Like these two, they were approaching this car because this man in the driver's seat was throwing bread for them out of the car. So, and even after this man drove away, a lot of people were trying to approach these birds to get closer and see them a little bit. And these birds, thinking they're going to get food, are just very slowly inching towards them, asking for food. And that's not good, because we have to make sure that Nēnē are not reliant on humans for food and they continue to support themselves and forage. But being in this parking lot is also very dangerous because it again increases road collision chances. So placing these do not feed signs and signs reminding visitors not to venture too close to them are ways that seem to be helping prevent this. It's also really important to drive the speed limit within the park and looking out for birds on both sides. To my main goals and tasks with my internship participation, I also got to attend an outreach table for Volcano school, and it was part of their career day. So they came to the park as part of their field trip. And there were several tables, including for the wildlife team. It was also felt like my career day. I got to see these different organizations that I otherwise would not have met and got to learn about. But for the wildlife team, half of it, the table was split for birds, for ʻUaʻu and Nēnē, and the other half was for sea turtles. There was another day that I got to join on a day excursion to the Kahuku unit. So here it is on the map in relation to the main park. It's towards the south. To look at, I was focusing on ʻUaʻu, and I was visiting the Hawaiʻi and Kahuku unit, which is part of the southwest drift zone of Mauna Loa. This is the only day that I got to visit Mauna Loa for my internship participation. The rest was on slopes of Kilauea. And this day's main goal was to confirm that these burrows were inactive and ʻUaʻu was not nesting, and removing the trail cameras from these sites. And the area is a much older lava flow and covered with cinders, so you can see this very drastic difference in terms of the colors of the dirt in the soil. And the most recent eruptions from Kilauea are blowing tephra and adding to this space and further vegetation and shrubs in this area. So it's very dry, and you can see these brown and red but they're all very crumbly lava rocks. They're very difficult to walk on. And on a particularly windy day, some of dust coming into your eye. I had a lot of fun. It was a really cool experience to visit this area. I haven't seen an environment like this before. So even get to see these inactive nests was a lot of fun. Outside of my internship, last summer I got to attend Hawaiʻi Conservation Conference and get the opportunity to not only support some friends and people from this school and my cohort, but also my undergrad experience from the school. But I also got to see people from my community college and my undergrad experience on Oahu, so that was really great seeing everybody, but also learning about the different research occurring on these different islands was really interesting. I also got to see my first research local species, which is the monoku, so I was very happy about that. I also got to visit Hakalā a couple of times, and on this particular volunteer trip, it was overnight. I got to stay the night at Hakalā for the first time, so that was really fun. And the opportunity, the main goal was to outplant ohia saplings and actually holding these ohia that I'm actually doing data analysis on was another way to connect with the work that I'm doing for my project. I also got to do a lot of birding and bird watching the next day. So I got to see Avian pull out Hilo for the first time. And up until this trip, I've only seen them on pictures or listened to their vocalizations on the computer. So being able to see them for the first time was a lot of fun. And this is before starting the Nēnē surveys. So seeing the Nēnē at a different location on the island was really fun. And you can see them in the middle. We created a space on the group photo to show the Nēnē. A little bit before that, I got to visit Haka to help place saw meters up for a classmates project. So that was a cool experience as well. When I mentioned that I got to listen to bird recordings and on the computer, it was on listening to spectrograms and listening with my headphones in a lab. So seeing the data collection aspect of my prior experience before that was also another great aspect. Overall, I think my 2 years in this program, I got to still meet my objectives and improve my writing and data analysis skills through this internship, but also through this program, while gaining more field and lab experience. And I got to actually experience bird banding and surveying and DNA extraction for the first time, or more extensively compared to before. So that was a lot of fun. I didn't mention this earlier, but I live in Volcano. So being able to connect with the town that I live in and work in the town that I live in and connect with the birds and the environment, a very fulfilling way to participate and complete my internship. It didn't feel like work. It was very important, fun, not entirely fun because you do have to deal with dead birds or even hear about dead birds at times, and that's not fun, but it's fun. So this connecting with birds and the environment was great. So I do appreciate the TCBES program and Lisa and my mentors, Eben and Kristina and Josie, for leading me through these excursions that I got to come and join, and the Hawaiʻi Foundation and this cohort. Thank you. Oh, yes, hi. The smaller predators like mongoose, they target eggs in the smaller birds, but the larger birds are getting more towards, we're targeted by feral cats and pigs. This kind of so that rainfall and weather definitely affects mosquito prevalence and the spread of avian disease, but specifically.
A little bit about how you bringing in bicultural and bilingual learning into your what you're doing.
Oh, yes. So as for the bilingual aspect of things, It's not with connection with my internship experience. It's more of a thing that I'm doing that contributes to my overall understanding of birds and ecosystems. I grew up in Japan and I am half Japanese and I speak Japanese. So my director of studies is looking at birds and bird terms and understanding and learning about birds in Japanese a little bit more. It's because something that I've studied, so I don't know how to talk about in my first language. And as for this summer, I'm going back to Japan to study, I'm going to visit a bird exhibit. So, just learning about birds in Japan, but also how a museum setting will explain something like this in a more formal setting. Really interesting. I'm also visiting New Zealand for the first time, just to understand predator control and their management strategies for conservation. Dissecting and that you know that spheres, the data analysis and survey. Experience. What part of that? Oh, I really enjoyed bird banding. So being able to actually hold the birds for the first time was really cool. Instead of just seeing a photo of how bright the green feathers are, actually touching how soft the feathers are and seeing in person how bright the feathers are. It doesn't really directly translate on a photograph very well, so seeing that in person was really good.